Okay, let's go on now on the production and function of white blood cells. So this we went through red blood cell. Now we are going to white blood cell, or in other name we call it leukocytes. So these are cells of the immune system defending the body against both infectious diseases and foreign materials. That's why these white blood cells are the one which they do fight against infection. That's why we call it the cell of immune system, because immune system is the one which is going to fight against infection. So the white blood cell are the one which are going to fight against the infectious diseases and other foreign materials. If, for example, any foreign material infection which can attack the body, the white blood cells are the one which are going to fight against it. So we have 5 different and diverse types of leukocytes which they do exist, but they are all produced and derived from multipotent cell in the bone marrow known as hematopoietic stem cell, as we saw in RBC. So we have the hematopoietic stem cell, or in other way we call it the pluripotent hematopoietic stem cell. This is the one which produces RBC, produce white blood cell together with the platelet. So we have all these stem cells, hematopoietic stem cell, which is responsible to produce this cell. Also, these leukocytes are found throughout the body, including the blood and lymphatic system. Now we are going through the structure function of the blood, and the next lecture tutaona lymphatic system. So now let's concentrate on the blood system. Though RBC, white blood cell or leukocyte, they are found both in blood together with the lymphatic system. The aim is to fight against infection. So the number of leukocytes in the blood is often an indicator of disease. For example, now if the leukocyte is above normal range, inakwambia kwamba nao kuna disease inayoendelea, there is ongoing infection. So this indicates there is a disease. For example, if the number of leukocyte raises or number of the leukocytes zikiwa chini, inakwambia kabisa now there is a disease or ongoing infection. There are normally leukocyte normal zinakuwa is 4 times 10 power 9 to 11 times 10 power 9. So above 11 times 10 power 9, it means there is leukocytosis. It means there is an ongoing infection which is going on, any disease. It indicates there is disease now. Or if there is a leukocyte below 4 times 10 power 9, also that is leukopenia. Also it is indicator of disease. Mainly leukopenia inakuwaga ni viral, but sometimes viral unaweza ukaikuta kuna leukocytosis with predominant lymphocytosis. So it make up approximately 1% of blood in the health of that. So in conditions such as leukemia, the number of leukocytes is higher than normal, and in leukopenia this number is much lower. So in leukopenia kuna kuwepo na low number below 4, and also in leukemia kuna kuwepo na leukocytosis. It means trying to fight against infection. Also we have these types of white blood cell. There are several different types of white blood cell. They all have many things in common, but are all different. One primary technique to classify them is to look for the presence of granules. We have these granules which allow the differentiation of cell into the categories, the granules and agranulocytes. So these granules are found inside the white blood cell. These are the one which do allow the categorization or differentiation of this white blood cell. That's why we have white blood cell which possess granules, and we have those blood cell which do not possess granules. We have the granulocytes. These granulocytes are cells which possess granules, and agranulocytes, these are cells which lack granules. So we have granulocyte. Granulocyte, these are the cells which they do possess granules. In other way, tunaziita the polymorphonuclear leukocyte. This sorry, I didn't explain more on these granules. These granules are found in the white blood cell. Any blood cell, white blood cell ambayo ina granules. In other way, tunaita nuclear. Ina granules kama kidot, tunakita granules kama grains kwenye cell, that is nuclear. So white blood cell ambayo haina granules, in other way, ambayo haina nuclear, inaitwa agranulocyte. But white blood cell ambayo ina granules or nucleus tunaita granulocyte. So now you're going to see the leukocyte or white blood cell ambayo ina granules tunaitwa granulocyte. Ina maana ni polymorphonuclear leukocytes. So leukocytes characterized by presence of different staining granules in the cytoplasms when viewed under light microscope. You may find one white blood cell in a lot of granules, a lot of nucleus. They are mostly visible in microscope. So these granules are membrane-bound enzyme which primarily act in the digestion of endocytosed particles. Also cell itself, it is cell yenyewe pia ina particles ambazo zinasaidia katika digestion ambazo zinaitwa endocytosed particles. They are responsible for feeding the cell itself. So there are 3 types of granulocyte. Granulocyte meaning the white blood cell ambayo ina possess granules, or white blood cell ambayo ina possess nucleus. We have 3 types. We have neutrophil, we have basophil and eosinophil, which are named according to their staining properties. Why imeitwa neutrophil tutaona, while inaitwa basophil, why inaitwa eosinophil. They are named according to their staining properties. So the granulocyte formation involve major changes in nuclear morphology and cytoplasmic content, which are the best known for the neutrophils. So this proliferative cell have large euchromatic nuclei and lack cytoplasmic granules. Here we said this granulocyte are the cell which involve a major change in nuclear morphology and the cytoplasmic content, which are the best known for the neutrophil. Zinakuwepo zina change, because even if there is infection now, you may just see, or if there is infection or any tumor or any cancerous things which is going on, you may see if there is any problem or changes in this neutrophil. That's why this proliferative cell have large euchromatic nuclei and lack cytoplasmic granules. Why? For example, if there is a cancer, once you take a tissue, now you visualize in the microscope, you may find the nucleus is so euchromatic. Unakuta iko ina rangi sana inayoonekana, na also inakuwepo ina lack cytoplasmic granule. That's why sometimes tunasema there is increased cytoplasmic nuclei ratio. So they differentiate into slightly larger polymerocytes, in which the first group of specific protein is synthesized in the larval endoplasmic reticulum and Golgi's apparatus. That's why we said in RBC there is no nucleus. At the same time, there is no protein synthesis in RBC because they lack nucleus, they lack granules. But in white blood cell, they have granules, they have nucleus. That's why we have all these endoplasmic apparatus. We have the Golgi apparatus. We have the endoplasmic reticulum, which are responsible for synthesizing the protein. And this Golgi apparatus, endoplasmic reticulum, sometimes they are responsible in synthesizing this protein. what this protein also, we need this cell for themselves to work and to be fed. So the protein are stored in large primary non-specific granules, which are large lysosomes containing acid phosphatase. So smaller secondary specific granules are formed in the smaller melanocytes, which are the last proliferative stage. So for the cell themselves to be formed, we need them to be in a proliferative stage, for them to proliferate, for them to divide, for them to make a lot of white blood cell to fight against infection. For the cell itself, it needs protein, inahitaji nutrient ambazo itaweza kuisaidia to fight against. So the nucleus is typically flattened or slightly indented on one side in the melanocyte. So we have also metamelanocyte stage. From the melanocyte, now we have the metamelanocyte stage. The cell size inapungua and the nucleus become heterochromatic. Heterochromatic kunakuwepo ni nucleus zipo nyingi lakini zinakuwepo in different color zake hazilingani. That's why heterochromatic. Ile ya kwanza ilikuwa na euchromatic nucleus ambazo zilikuwa kwenye color inayofanana. So inakuwa ni horseshoe shaped and the protein synthesis almost stops. So protein synthesis, it stops at this stage of metamelanocyte. At the neutrophil now inakuwa released from the metamelanocyte. Now the neutrophil is now released, of which is matured now. The nuclei become first heavily intended and subsequently segmented into up to 6 lobes, characteristic of a mature neutrophil. So we found this neutrophil now is matured into 6 characteristic lobes. Hiyo utajua kabisa now this is a matured neutrophil. But this process, almost like in RBC, it takes place 7 days for it to be matured, of which 3 days spent proliferating and 4 days maturing. Kwa inachukua siku 3 kudivide na inachukua siku 4 kuwa matured. So the neutrophil may then be stored in the marrow for further 4 days depending on the demand before their final release into the circulation. So this eosinophil also and basophil pass through the similar sequence, but their nuclei do not become as irregular as that of neutrophil. So we have this we have neutrophil ambao tumeziona ziko released from the metamelanocyte. So we have other cell of the white blood cell granulocyte. Tumesema kuna neutrophil, kuna eosinophil na basophil, na zenyewe zinapitia stage ile kama alivyopitia neutrophil, but the difference ni kwamba nuclei zake haziko irregular kama zile za neutrophil. So basophil nuclei ziko bi or trilobed, lakini zile za neutrophil tuliona zina 6 lobe, lakini za basophil zenyewe zina lobe 2 au 3, but it is hard to see because of its number of coarse granules which hide it. So also in basophil they are characterized by their large blue granules. So kwenye basophil granules zake ni za blue, that's why ikaitwa basophil. Tulisema kwamba zimepewa majina yake kutokana na color of the staining in microscope once you visualize. So the eosinophil yenyewe ina lobu 2, but with a neutrophil nuclei ina lobu 3, ina lobu 6, na basophil nuclei yake ina lobu 2 au 3, lakini eosinophil yenyewe ina lobu 2 tu bilobed. So the cytoplasm is full of granules which assume a characteristic pink orange color with the eosine staining. So now you are supposed to know here in the eosinophil, ukikuta hata siku mtakayosoma pathology hii hapa, mwelewe hapa kwenye pathology siwasumbue. Ukipewa slide yoyote ukakuta white blood cells za rangi ya pinki, ujue hiyo ni eosinophil. Utamwambia eosinophilia, na kama ni eosinophilia utajua kabisa infection ipi inaleta eosinophilia. Na ukikuta blue granules au nuclei ya blue utajua hapa ni basophil. Utajua ni infection ipi inaleta basophil zinaongezeka. in pathology na kwenye clinical laboratory. So neutrophil also known as a polymorphonuclear leukocyte. Polymorphonuclear ni nuclear nyingi, that's why tukasema ina lobu sita. They have multilobed nucleus which appear like multiple nuclei, hence the name polymorphonuclear leukocyte, as I said. So the cytoplasm may look transparent because of fine granules that are faintly pink in color. So that was about granulocyte. Ziko tatu ambazo zina nucleus. Kuna neutrophil yana 6 lobe nucleus, kuna basophil yana 2 to 3 lobes nucleus, and also you have neutrophil. The basophil nucleus yake ni blue granules, eosinophil ni pink granules. So na nyingine utaitofautisha kwa sababu inakuwa na nucleus nyingi, 6 lobes.